Hi class, and welcome back to another asynchronous lecture for Psych 101, Intro to Psych. Today we're going to go over Chapter 3. One thing to note about Chapter 3, the biology of the brain, is it is very detail-oriented. While when we talk later in the semester about larger picture, more abstract things, maybe we'll talk about relationships, we'll talk about how we think, we'll talk about learning. Here we're talking about some very specific concrete things, so neurons, neurotransmitters, how the brain works. So this lecture is a bit different than some of the other ones later on, where we're talking maybe more about theories in which we can't exactly see but can test. Here we're very specifically talking about the things that we do see in the brain, right down to the molecules and structures, cellular structures of the brain. So recognize that if you're somebody like me who isn't exactly the best at biology and things like this chapter might take a little bit more work for you than some of the other chapters. But that's just an FYI, because I'm sure there are some of you, some of my health majors, public health majors, who are really going to get a lot out of this, I think, maybe more than some of us psych majors. So with that in mind, let's go ahead and start with the announcements. Nothing too big today to bring up. Just keep up with your materials. Make sure you're watching this video and reading the lecture, watching this video lecture and reading the materials before the next exam. Before the next exam, obviously, but also before you take that quiz. Make sure that if you do have any questions regarding the class, the reading, the asynchronous lecture, anything, you can always stop by office hours. I had a couple people in some of my classes bring up things that they maybe weren't doing so well in, or had some kind of trouble understanding in the video lecture or in the book. I encourage you guys to talk during class and ask those questions. Hey, I wasn't really sure about this. Can you help me? Or can we go over some of this? Because that's hopefully what the in-person class is for. Not only that, you can always stop by my office hours, also by appointment, send me an email, and I'm happy to chat with you guys about what it is that maybe you're having a little trouble answering. That's the worst part about these video lectures, is the fact that I just sit here and ramble and don't get to check if I'm making any sense or not. Hopefully I am, but if not, please always feel free to call me out. Speaking of the fact that these are the video lectures, I wasn't sure if I mentioned it to my intro classes, but one useful thing that YouTube does is, one, it should be putting up auto captions for me, which are not going to be correct all the time, and they're probably going to say some dumb things when I talk about different psychological nuanced words and kind of definitions that maybe the closed captioning software doesn't understand. But the other nice thing that you can do is you can increase my speed, my talking speed, and watch this video in about half the time if you go to 1.5 speed. Some people really enjoy watching the videos a little bit faster and getting that material. I know I watch a lot of YouTube videos where people are talking, and sometimes I like to speed them up as well. Just know it is an option for you that you can always go back, rewind if you're missing something, slow it down. But I recognize a lot of students might actually like this feature, so it's worth mentioning. With that in mind, let's talk about what we're actually talking about today. 3 minutes in, it'd be nice to get there. So we're going to focus on 3 specific areas. We're going to talk about, firstly, the building blocks of the nervous system. So what is the nervous system and what's it made up of? Then we'll talk about 2 branches of the nervous system, the peripheral and the central nervous system. Now, with the nervous system, this is a core component that doesn't just involve the brain. So the nervous system is not the system in which you are nervous, right? That will be part of the nervous system, but the nervous system is actually going to be our system of nerves and nerve endings, as well as the brain, in which case this is going to kind of explain who we are and how we feel and sense the world around us, right? It is our nerves, it is our senses, even. And this system is going to be what runs us, right? This is going to be in the same way our operating system. And while we have the hardware, the rest of our body, we need the software to be active to get things going. So the nervous system is technically, by definition, a network of cells that carries information to and from all parts of the body. This is an information highway. Right? This allows us to gain information about what's going on outside of the body right, in our senses and inside of the body, right? We know when we have headaches or maybe our stomach doesn't feel good, we can gain that information from there. Now you can see there's a ton of different nerves, nerve endings, just parts of the nervous system. We do not need to know all these different nerves. We are going to just be very kind of general regarding this information. So do keep that in mind here. But this does allow us to gain information about what's going on inside and outside of the body. But the interesting thing is the nervous system can be broken down into a really simple set of 3 functions. And they are as follows. Just like with our brain, we can't necessarily open it up and figure out what's going on very well. In the same way that as your book talks about, you might be willing to take apart a piece of hardware. Here I have my clicker that I use, a remote, something like that, and see the inner workings, the circuits, the wires, etc. We can't really do that with the brain, especially because, as we know with things like this, it's often hard to bring them and put it back together to begin with. So the 3 basic functions of the brain, the nervous system, are to 1, receive inputs from the environment. Now that can be our senses, what we hear, what we see, what we touch, what we taste. There are other senses, We have a sense of time, we have a sense of direction, we have a sense of balance. And all of this input, helps us understand where we are, what we're doing, what we see, what's going on. It orients us to everything. So 1, we receive these inputs. 2, we process these inputs and we integrate them with our past experience. If you smell something, you go, what's that smell? Why does that smell familiar, You might integrate what you know, about that smell, or recall or think or memorize or remember about that smell, and integrate it with what you know. If you see a bear, coming towards you in the woods, you are going to integrate that with past experience. Go, I know what creature that is, and I don't think, from my knowledge of things I should be doing, I don't think I should be standing here if that bear is going to chase me. And so what that does is allow us to generate outputs, and we call those behavioral responses to the inputs. Technically, we say behavioral responses. However, we know neurons are active when we think, so it is just kind of responses in general. You could call speaking a behavioral response, speaking your thoughts, but also, even when you're sitting there dead still and not doing anything, you are still, your brain is still working, you're still thinking, your heart's still pumping, your blood is still going through, you're still breathing, and all of this is still wrapped up in the nervous system. So it's a really simple sounding system. Take what you see, feel, hear, touch, etc. Take all that information, dig through it, figure out what you know, what you don't, what seems important, what doesn't seem important, and then figure out what to do with it. That's a pretty big task, and we can do things super fast. We can talk about some of the things and how we can do these things so quickly. So the basic building blocks of the nervous system, you've probably heard of this before, are what we call neurons. Neurons are specialized cells within the nervous system that both send and receive signals and messages within that system. That's a big definition. So what do we mean by that? Well, we've got neurons. And neurons are the types of cells that we work with in our brain. And they have 2 functions. They have an input, which is over here, and they have outputs, which are over here. So they can send messages with the outputs, and they can receive messages with the inputs. Now, at the edge of our body, we have nerve cells or nerve endings, which are the ones beneath our skin that feel things like touch, heat, other kinds of senses. Maybe our eyes have specific types of we'll talk about them later, but rods and cones that we can see with. And these are our basic sensory organs, touching, feeling, tasting. But once we get that signal here, on the edge of our finger, we've got to run that all the way up the body to our head to know what that is, If I'm, doing something annoying and if you ever do this to somebody, you pretend to just touch them really lightly on the back of the neck, They're starting to figure out what that is, and that is going to be that kind of input, the receiving that message from either the outside world or another previous neuron in our chain, and sending that message onward if it needs to be sent onward. That's a lot. I definitely went into that pretty heavy, and we'll talk about and we'll unpack a lot of what that means here in the coming slides, but recognize that neurons are going to be the bread and butter of what we talk about. Something else I do briefly want to mention, right? Our brain is not simply made up of neurons. It is also made up of what we call glial cells. And glial cells are the types of cells that actually direct the growth of neurons, provide nutrients to the neurons, and help repair and help damaged neurons from being destroyed. Apologies, I didn't realize you couldn't really see the bottom of that, but it's fixed now. So, neurons. This is kind of the general look of what a neuron is, but I have a crappier, drawn-out version here that I think is very useful to talk about. So, what we have here is, right, more or less a neuron. So here's a version that's a little bit more stylized. I'm not going to use this. I'm going to go back to my other version. But neurons generally are broken down into a couple different parts. So we have this kind of large part over here. We have the longer portion here, which we can see these kind of yellow things, which we'll describe, which are not actually on every cell. And then we have down here this kind of other aspect. So I like to show that picture, so that way you can see part of what a neuron looks like, but also recognizing that my drawing skills maybe here are going to be a little bit clearer. So the cell, first of all, of a neuron has what we call the body, or the soma, S-O-M-A. This is not the same word as the online software you're going to be using to take experiments or sign up for experiments, right? Sona with an N. This is going to be soma with an M. And the soma, or the cell body, is again kind of this major player in the neuron, because it's going to have the DNA, the kind of structure, all of the important parts of the neuron cell. It's almost like the head, the head area of it. And attached to this soma, we have what we call dendrites. And dendrites are, I like to think of them almost like roots. So dendrites are like roots, and if you can actually see over here on this side of the screen, on the right side of the screen, here are dendrites for the next neuron in the chain. So these neurons are going to be stacked, you think of a pen like this, kind of like one after another, except maybe more in reality. At the base of one neuron, the end of this guy, there's going to be a couple different neurons sitting right around it that are also going to be used. So dendrites have a very important job. They take information from the previous cell and send it to the soma. And the soma is going to make a decision. The soma is going to make a decision whether it wants to fire or not fire. And that's all our brains, or neurons I should say, do. Neurons have two functions. They can fire or they can not fire. So go or stop. Well, when the other parts of this, which will help elucidate what I mean by what I say, is the axon. So that long kind of path, here it's straight, in other places you see it's squiggly, that is going to be the axon. And this is going to be you can think of it kind of like the highway, sending information down towards the end of this. Now, some cells have these gray, as I have them here, blobs. They almost kind of look like sausage links on the outside of the neuron. So if I go back for a second, these guys, right, these are the myelin sheath. And the myelin sheath is actually a bit of fat or myelin around our cell, which helps speed up the transmission process for certain types of cells. Now, I'm not going to go into that too much, but the idea is that a myelin sheath allows cells to be even quicker than they are. And a reason we might have that is for our motor cells, for instance, or our motor neurons. Those we want to be very, very quick. We want to send signals from our brain to our hands as fast as possible, to our legs as fast as possible. And because those are so far away, we can actually speed up the process using myelin sheaths. So a myelin sheath is going to act as kind of almost like like a speed boost for sending signals along the way. But as we work down the body of the neuron, we get to what we call axon terminals. So axon terminals are these circular portions right here. And within the big circular portions, I should say, these bigger circles, and within these, we have these smaller circles that we call vesicles. And vesicles are going to hold neurotransmitters. So vesicles are almost like buses or planes in the sense that they kind of pack people, in this case neurotransmitters, in to their little vehicle that they are. Vesicles are vehicles that submit or send this stuff, our neurotransmitters, into what we say a synapse, or this kind of break in between the vesicles here at the axon buttons and the next dendrite. That's a lot, and I know that's a lot. So let's try to break some of this down to make a little bit more sense. So we know how this looks, right? Is that, and one way to think of this is from left to right, this information is happening. You can almost think of information happening to the brain like a wave, right? Is that, right? It kind of goes, like the wave continues this way, right? And it's like if we had a group of people and we did the wave, we know the wave continues on, but those cells, right, stop eventually. And the way that we can think of this, right, is that a signal is going to come in from the dendrites. It's going to go to the cell body. The cell body, the soma, is going to decide, hey, there's enough signals that say go, that I'm going to go. The signals then send that down the axon, possibly with a myelin sheath to go even faster. And that information goes down to the axon terminals. The axon terminals kind of get, the vesicles in the axon terminals get pushed out into the synapse. And in the synapse, those neurotransmitters spill out, and then they bind to the next set of dendrites over here, going back to square one on a new neuron. So a big kind of chain of events. So, right, we start our wave up here, and we move it down to the end. So how do these communicate with each other? By transmitting electrical signals along the axons and then sending chemical messengers or chemical signals into the synapse. I'm not going to go into too much detail about this, but the process is known as a neural impulse, and it's an electrical process guided by things like sodium and potassium ions. So if you took chemistry in high school or college, you know about ions, which are positively or negatively charged, usually, I think usually just like elements, right? So a potassium element or a potassium molecule that maybe has one extra electron is going to be positively charged. But if there's no information traveling through a neuron, the axon terminal is what we call at a resting position. So it just kind of sits there. So it just, right, nothing happens here. If, though, there is something being submitted, if there is information to be transmitted, the ion channels, which are more kind of like lids or gates on the axon, on this portion down here, right, these axons, are going to open up, and it's going to allow these different ions to rush in, which creates a temporary positive charge, and we'll again, I'm getting into too much detail too early. So here we go. We've got the features of a neuron. We know, right, we've got our dendrites, we've got our soma, we've got our axon, we've got our axon buttons, we've got our vesicles. Here I actually don't have a myelin sheath, as you see. So this one's going to be slightly slower than the other ones. So how do these cells communicate? Well, they communicate using what we call action potentials. And action potentials are an electric signal that we conduct along the axon to a synapse. So from here, down here, to the end. These action potentials are what we call all or nothing. So you can think about them like a gun firing. If you have a gun, don't try this at home. If you have a gun, it's going to have a trigger. And you can push on the trigger a little bit without it going if your gun is set up correctly, right? It needs a couple pounds of pressure. So think about it like this. Over time, if This is my, this will be my trigger. How do I want to do it? This will be my, so I have a trigger and I need to pull it to, right, shut it and make this neuron go. Well, I keep getting some signals, this electrical signal that says, hey, it's getting a little bit stronger. It's getting a little bit stronger. It's getting a little bit stronger. And at some point, I hit what we might call a point of no return or an action potential, right? That I've hit critical mass or excitement. And at this point, click, it's done. So I guess like this would be a better example. At some point, I can push down, push down, push down, and then eventually, right? Now there's no going back, right? It's gone. Well, it's done. So I can push a little bit, right? And that's fine. Nothing will happen. But if I hit that point of no return, that action potential, then the cell fires. So recognizing that, yes, this electrical signal is slowly changing, and then boom. So how does that look? How does this gun firing or clicky pen thing work? Well, we know that these cells are polarized. As mentioned before, these are more negative on the inside. And what we have are these channels or selectively permeable membranes. So they are permeable, they can let things through, certain things through, and they're selective about it. So they only let through what we call, right, positive ions. Right, so these channels here, right, we have a negative charge inside the cell. Outside the cell, so in your brain, outside of each neuron, in that cerebrospinal fluid, is going to be a slightly positively charged liquid. And we're not talking like it's going to shock you. We're talking like less than, right, very, very tiny amounts of voltage. And your brain is really salty, right? It has a lot of sodium in it, right? That's why sodium is important. It also has a lot of potassium in it, and potassium ions. And so what happens is that if we add a little bit of excitement, so right, if you're like my dog, you might, right, a good example is my dog gets really excited and there's I don't want to say that word, TREAT nearby. And what happens is that kind of looks like these signals being sent saying, hey, we should go. And then we add a little bit more excitement and hey, we should really go. Now we should definitely activate. And now we're closer to that action potential. But eventually what happens is we can't let that excitement go anymore, right? This dog is clearly at its wit's end. And right here, we see these polarized cells sweep down, which opens our ion gate channels and lets in these positive ions now. So these negative ions down here that were in here now kind of get pushed out for a second. And this happens like a wave, right? So it starts up here, it moves down, and then we have our first ion gate open. And when that ion gate opens, we're letting more stuff in to rush in further. And then the next one opens, we're letting more stuff in to rush in further. And then the third one opens, right? But there's many down the line. And now we're just starting to create a stampede, you might call it, right? Is that eventually they're all, right, going in this one direction down the axon, towards the axon buttons. So to summarize that, we've got our dendrites up here, which are getting these signals from a different neuron, right? And here's the end of this neuron, so you can imagine other neurons, right? Here are some axon buttons, right? Right here are some dendrites that are going to get that signal. The dendrites get our input. That information goes to the cell body right here, and our action potential occurs. So eventually, right, or I guess better done, we've hit our action potential. The gun has fired. And now that action potential can't stop. It goes all the way down the synapse. Now, for what it's worth for people who are interested, here we again have our myelin sheath. What the myelin sheath does, it allows only specific areas to have these open gates over here, which helps speed things up. It's like creating the right amount of traffic, not too much to kind of push it down. They're faster. And so when these action potentials get all the way down the axon to the axon buttons down here, here's the one that we're going to talk about, which is right next to another dendrite. These neurotransmitters here are going to be released at the axon button by the vesicles. And we'll talk about that here in a second a bit more. But as you hopefully know, these cells can't fire the whole time, right? After this has happened and we have all of this kind of positive in here, we have to repolarize. And then we call this a refractory period, which you may have heard of in other scenarios. But in a refractory period, and what we're talking about here specifically, is the idea that we are kicking out those positive ions that don't belong there, right? We know the cells are negatively polarized. They have a slight negative charge. Well, after all the positive ions come in, that's what sets this all off. We have to kick them out, right? Because we want to get back to normal. So all of our positive ions get kicked out, and our other, excuse me, man, I forgot how difficult this is to explain. And then we are trying to get back to that normal level negative and back to our kind of resting state, pre-action potential. So this kind of all floods out, and then eventually we end up back at normal. So great, we have now had our single neuron fire, and that is how a neuron fires, in recognizing that there are billions of neurons in the brain, which are all used at some point to help us understand something. So we've now talked about the super duper duper micro for a second, and we're going to keep doing that. But one thing I want to talk about is, what does this mean in terms of like what is actually going on? So what does this look like? Well, we receive an input. So we put our hand on a hot stove, and all of a sudden that triggers something, right? That triggers the first action potential in a series of action potentials. Dumb joke, but I love it, right? Why was the neuron scared of exceeding negative 50 millivolts? Well, because he didn't want to get fired. He didn't want the action potential to fire. Haha. Anyways, I'll keep my day. Actually, this is my day job. Shoot. But anyways, moving on from there, that signal travels via nerves and the neurons in the brain. Each signal goes all the way through, right? This process back here. Right. Each signal has an action potential that fires, and that goes all the way up our brain, all the way up and back down after it is interpreted by our brain as something, hey, this is bad. And so that response is sent up and back down very quickly. But how fast? Well, neurons are fast, very fast. Myelinated neurons can actually send signals at speeds between 70 and 120 meters per second. That's 156 to 270 miles per hour, right? So imagine that there's just one neuron that needs to get from the base of your foot to the top of your head. That's still incredibly fast, but we're talking about many, many, many of these, right? And they're not that long. Well, right, most of them are about 1 micrometer in diameter, so less, I believe, about the size of a width of hair, if not even that, and about 1 millimeter in length, right? So super duper tiny, super incredibly tiny. And those are moving at over 200 miles an hour, some of them. And that's how quickly this is going from base, right, from bottom of your foot to the top of your head and back down, or wherever it might be. Though the spinal cord takes a lot of the longest neurons and kind of holds them together. It mostly are motor neurons. And the longest neuron, which goes from the base of the spine to the bottom of the toes, is actually over 1 meter long. So that's still only a 70th of a second to get from the base of your foot, right, if you step on a Lego, all the way up to your spine. And the rest of the way up is still nearly as fast. Now, these are, again, myelinated neurons, which are slightly faster, but again, it's not like regular neurons are only going 10 miles an hour or anything. They're going still very, very fast. So while it seems like crazy when we talk about all that we just, right, the last 20 minutes of lecture where I talked about first these signals come in and then they have to be determined if there's enough signals to create an action potential to send it down the line to make the vesicles sent out and to do the process over again. That is happening so incredibly fast. It's just it's mind-blowing, and it's something that every time I teach this lecture, I think it's almost unbelievable how fast that works in our brain. And so with these action potentials at the end of the line, these vesicles here, what they do is they kind of pop out almost like a balloon and let out the neurotransmitters here, which we'll talk about more in a second. But we know that these electrical signals from these action potentials cause a neuron to fire. And when that neuron fires, this electrical signal with all those ions rushing in travels down the neuron to the axon terminal where, as we said, the vesicles are sent out. But we know that as the neuron fires, the vesicles head out. These contain the neurotransmitters that start that chemical reaction process. So how do they get there, though? Well, what happens is, more or less, these vesicles package up the neurons together. So this is a pseudo-colored image via an electron microscope of the terminal button, that green area at the end of that long axon, Our axon terminal ends at that terminal button. And here we actually see, like this, this button being opened, as we can see in this photo here, It kind of popped it open. And so what you see here are the vesicles in blue and orange. And so we have vesicles down here, but we haven't even seen neurotransmitters. And again, we're talking about 100 micrometers. So this is a rather thick end of the neuron, because we said they can be as thin as, 1 or 10 micrometers. But each vesicle actually contains about 10,000 neurotransmitter molecules. Each one of these tiny blue cells or orange cells over here contain over 10,000 neurotransmitters. We're talking, these could be all dopamine, these could be all serotonin, these could be all GABA, or some of the other ones we'll talk about. I don't know about you, this stuff gets me kind of hyped because this is just incredible the way that we operate, The fact that every time I'm deciding to say something, I have this stuff going on in my head, in my body to make this happen. So the reason that I bring this up and to make this very clear is we've got our electrical signals, which have got us to the point where we now are sending out our vesicles, which are full of neurotransmitters. And one way we can think about this is kind of like that axon terminals are a bit like airplane terminals. So here's an axon terminal, We've got some vesicles down here. We've got some ion channels up here, which we don't really need to care about. We've got our next different, we call this a postsynaptic cell, or after the synapse, so the next one in the line. And I like to think of them kind of like airplane terminals. And the reason I say that is because neuron transmitters in our axon terminals, they basically get packaged up into those vesicles. And those vesicles are the ones that are getting sent to the synaptic cleft to take the next neuron. So our neurotransmitters are kind of like passengers hopping into the vesicles that are planes. And those planes take off and they get to their location. Now, the nice thing about that metaphor is it kind of puts things into perspective, but don't let me fool you. The vesicles are maybe the worst pilots ever, So once we get into the synaptic cleft, the vesicles let the neurons go. So it's not the case where these vesicles stay open. These neurons get let out. So more kind of like, all right, we're out of here. We dump you out. This is going to be the worst pilot ever because he takes you halfway to your destination and throws you out. So I do want to spend some time recognizing that aspect of this, that it is not this clean and simple picture, This is a great picture because I think it really shows just how much is going on in each different neuron at a given time, and that we haven't even seen the neuron or neurotransmitters here, right? You don't see any of the ion channels or anything like that. This is still something we're working on to this day to fully understand. Okay, so let's talk about neurotransmitters for just a brief minute. So neurotransmitters are, right, these passengers on these vesicles or planes. And the neurotransmitters are now the chemical messengers that transmit a message from one neuron to another. So, keep going back. Blah, 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 blah. That'd be smarter if I'm going to pause this when I go forward to make more sense, I guess. Okay. So all the stuff that we're talking about here with the neurons, right, where these are kind of, right, being, having these ions come in and out, these are all going to be an electrical signal. All of this is based on electric, right? The millivolts in our brain are changing from negative to positive and sending that signal down like a wave. So we have this electrical signal, but we have a chemical signal as well, and these are our neurotransmitters, as I jump back to the right slide, which I now am on. So the point of that is, while we are getting this electrical signal that's sending our neurotransmitters out, our neurotransmitters are messaging from one neuron to another, almost like each neurotransmitter is a letter being sent out to the next neuron, right, through a postal service or something. But what kind of messages do they send? Well, we know they send a chemical message, but those signal types come in one of two flavors. They come in stop or go. As I said at the beginning of this lecture, our brain and our neurons operate on one set of rules, and that is, should I go? Should I fire? Or should I wait? So with neurotransmitters, we have two types of signals. We have excitatory neurotransmitters, and right, what is going to be a neurotransmitter that makes something excited? Well, it's going to send a signal to the next neuron, hey, you gotta fire, right? Go. Excitatory neurons, right? If I get my dog all wound up about going to the PARK, which I cannot say because she'll get all wound up, right? That is going to be, right, me acting like an excitatory neuron to my, right, the next excitatory neurotransmitter to the next neuron, my dog. But we also have inhibitory neurotransmitters, which send a next signal to the neuron to stay at rest. Hey, stop, right? Relax, I might say to my dog. And that is going to, with enough of those signals, tell you to stop. So although a given neurotransmitter can only say go or stop, there are many types of neurotransmitters, which I want to jump back to in a second. But really quick to recognize is that neurotransmitters are hitting your cells many times over, right? It's not just a single neurotransmitter. It's not whichever one gets there first. It's a lot in a lot in a lot. So as I switch back to this another slide here that we talked about before. So this is going to be what is going on up here in our dendrites, right? These are going to be our neurotransmitters sending signals up here to let in some of those positive ions. So as we see, right, a couple neurotransmitters start this, and then as we get more and more, we see this flood happening. So what does that look like? So jumping ahead a little bit, we as I said, we have these two types, right? Stop or go, excitatory, inhibitory. Flip those. So go, excitatory, stop, inhibitory, right? Inhibit from happening. But what is happening is these neurotransmitters are going to these channels, these ion channels at the dendrite and saying, hey, open up, right? We can see this guy is open down here, right? He's letting stuff through, or we can see, right, it's being closed. Okay, so some of these are naturally open, some of these are naturally closed, and as our neurotransmitters bind to these receptor sites, we'll call them, right, they are going to fit certain types of receptors. So maybe only serotonin works on this type of neuron here, or this neuron here is only focused on that. So if another neurotransmitter gets there, it's like you trying to trying to open another door with a different key. You're not going to get in, right? Only certain keys fit certain locks. And there are a lot of keys, different neurotransmitters that we can talk about. I'm not going to spend too long talking about any given ones of these, but the three I think are really worth mentioning, and I consider some of the most important ones that you can take out of this class are dopamine, norepinephrine, and serotonin. And dopamine, to start off with that one, is related to mood, sleep, and learning. Some effects of behavior include increased pleasure and actually suppressed appetite. But what we think of dopamine as, and in a kind of general way to think of dopamine, is a happy chemical. So dopamine is a chemical that is going to be released in your brain when something good happens. So when you're rewarded for something good, right, and you're learning to be rewarded for that, you are going to get, or you're going to have dopamine released. If something good is happening to you, maybe dopamine is being released. And this is going to help, right, to minorly, in a way, affect how you feel. So when you see, right, somebody you love after a vacation that they get back on, say it's been a while, your dopamine levels are going to shoot up when you see them. And that's going to be related to increased pleasure and being able to see the people you love. Actually, I'm going to skip down to serotonin, another one that many people have heard of. This is oftentimes considered the happy chemical in the brain. And this is a very mood-focused chemical where it seems to be the case where more serotonin leads to more positive moods. You also notice that it suppresses appetite, which is interesting, but we will talk about that later. And the third one is what we call norepinephrine. Norepinephrine has another name that people often know it by, and that's adrenaline. Norepinephrine and adrenaline are basically the same thing. And when we run into a situation in which something scary or bad is happening, we might have a release of norepinephrine. If you've ever jumped, right, at a jump scare and felt your body for a second afterwards, you might feel, right, that kind of jolt going through your body. And that is your body sending those neurons, those neurotransmitters and telling your heart and everything else, hey, time to get going. Right? And so you can almost about half a second later, if you're really paying attention, feel your body's blood pressure jump, your heart rate increase. You can feel it all the way down your body if you're paying attention. And those are going to be some of what are going to be affecting our brain, but not the only ones. But those are the three I would like to take a moment to talk about here. And those are the ones you should probably be a little familiar with if I were to ask a question on them. Hint, hint. So another question, or another thing to mention, is that we don't, right, when these neurons are done, or these neurotransmitters are done, they don't just disappear. Some of them go away, get repackaged, and put back in to our body again, up to our axon buttons. So we recycle these neurotransmitters, and that after sitting in the synaptic cleft here, they either bind to the next neuron, which in this picture down here is going to be looking like this, but then sometimes they get reuptaken. Or if they don't, they can get washed away and right off they go and usually repurposed for something else, or the glial cells might clean them up and try to get them back in circulation. But one way to think about this, and maybe you have heard of this, is a brand of anti-anxiety and or it's not a brand, it's a type of anti-anxiety and type of antidepressive medication. And things like Lexapro, if you've heard of that, Prozac, these operate on what is called, or they are what we call SSRIs, selective serotonin reuptake inhibitors. So, what do we mean by that? Well, selective serotonin, we are selectively looking for only serotonin, and what we are going to do is we are going to stop or inhibit the reuptake of this. So, what happens? Well, we shut down the reuptake things and we force these chemicals, these serotonin molecules, to sit out here for longer. If they're sitting out here for longer, they have a higher likelihood of being put into our next cell. So what we're doing is, instead of saying, hey, drop it, right? Like, okay, you know, whoever binded to their neurotransmitter, they did, we're done, come on back. It says, actually, let's leave them there for a little bit longer. So people with depression are getting a longer chance taking this drug to have the selected, or the serotonin that's been chosen, selected by this chemical to stay in that synapse. It doesn't let it go up as fast. It lets it sit there for longer, which is really cool, because if you are depressed or have high anxiety, we know having more serotonin in your body is going to help you. It's going to decrease symptoms of anxiety. It's going to decrease symptoms of depression. And so one way we can keep that in the body is we can force it to stay in these synapses for longer and not jump into right back up, to allow it to kind of marinate in our head a little bit longer, is one way we can think about it. Okay, spent a lot of time talking about neurons, spent a lot of time talking about neurotransmitters. I very much encourage people who do have questions to this to come to office hours to ask questions during class, because this is a very, very detail-oriented chapter. But off we go on to other things. So, what happens to all those neurons? Well, we've talked about the basic building blocks of these, but now we're going to go through the nervous system itself. So the nervous system is made up of glial cells, of neurons, just kind of sensory input cells, and motor cells for output as well. But we can divide this into two kind of sides of this. We can talk about the peripheral nervous system and the central nervous system. So while neurons are organized into groups called neural networks, the peripheral nervous system is the system without not dealing with bones. And right, we know bones are not part of the nervous system, but the peripheral nervous system is regarding things like muscles and kind of autonomic or kind of automatic processes within the brain. You can also think about peripheral as being kind of outside the center. If you think about what your peripheral vision is, right, I can see my hand here and I can see my hand here. But they're not in my direct cone of vision. They're at my peripheral. So this is going to be the part of our brain that maybe isn't the central focus. And if you look at what's in the central nervous system, it makes sense, right? Our central nervous system is our brain and our spine, that kind of dead center down the middle. Our peripheral nervous system is all the other stuff. So as we'll see here, we'll talk about a lot of things regarding that. So here's actually an example of the central nervous system. I know it says peripheral up here, but right, the central nervous system has our cranial nerves over here. It's got our spinal nerves. It's got our brain, right? And the peripheral nervous system transmits information to and from this guy, this central nervous system right here. This literally keeps us in touch with the world in terms of sensory and motor functions for the body. So what does that look like here? That is going to be this scary skeleton of a guy, right? This stuff all over here, and I can't do that, right? These, and these, the kind of straw portion of this straw man over here is what we're talking about with the peripheral peripheral system, because it's our sensory functions and our motor functions. So let's first talk about the somatic nervous system, or portion of the nervous system. The somatic portion, soma means body, right? S-O-M-A, we talked about this with the cells, right? Soma meaning kind of the body portion of the cells. Right, the somatic nervous system carries sensory information and controls the movement of the skeletal muscles. I said that this doesn't deal with bones. This deals with the movement of kind of the limbs and such, right? So the soma is going to carry this information for the body. And it works on two ways, as we've kind of said, on a sensory pathway. So what we sense, what we feel in our fingers all the way up to our brain, or the central nervous system where it's in the spine. And then we can talk about our motor pathway from the central nervous system back down. So, right, this cat here is probably going to touch this little fluffy duck, and he's going to feel its fur, and he's going to go, "Ooh, what's that?" Right? And then eventually this brain is going to say, "Hey, touch it, it's fluffy, it's nice. And then right, that motor pathway is going to be implemented here. So the sensory pathway is going to the brain, right? Because it's the sensory, right? What is it doing? It's sensing. And to sense, we have to send the information to the brain. And then the motor pathway down the other way from the brain to the muscles to move us. So that's the somatic nervous system, the part of this that is going to focus on carrying information and controlling the movement of our body. Now, the autonomic nervous system is where people can get really confused. Somatic nervous system, pretty easy. It's the body. It's the soma, the body. The autonomic nervous system carries messages between the central nervous system and our organs and our glands. So, right, all the stuff kind of in here, right? It goes through the spine, but what is it that's going through the spine and talking to the heart, and talking to the lungs, is talking to our spleen or what have you? Well, these are going to control things that we consider normally outside of our conscious control. Non-conscious, oftentimes, right? They are not within our conscious. Sometimes we can never even make them conscious, right? You cannot directly control your heart rate without doing other things, maybe thinking of things that scare you, right? You can't just look at somebody and cause your pupils to dilate or your blood vessels to expand or contract. You can't focus really hard and start digestion, right? These are things that we can't normally control. But why would we need to? So all of these things, right? Our blood pressure, our pupils, how we digest things, how our organs are working. It's not really useful for us to spend any mental energy on that. Our body can do it by itself, right? We can maybe activate it normally, right? If I say now you're manually breathing and now you actually have to take effort to breathe. Sorry, I have to do it. It bothers people, right? Because now you're forced to do it. And unless you stop thinking about it, you have to be able to think to breathe. So if you are, I do apologize. Take a break and go think of something else, go read something online, and eventually you'll stop trying to breathe. You'll stop being manually breathing. Okay, so recognize that the autonomic nervous system, though, comes down to two parts. So within the autonomic nervous system, we can come down to two different things, the sympathetic and the parasympathetic nervous system. What's funny is I almost always get these confused because they seem backwards to me. The sympathetic nervous system, when I think of sympathetic, I think of sympathy. I think of, feeling bad for other people. Right? I think of like not being happy, maybe being sad for somebody or upset. What the sympathetic nervous system is it's the fight or flight response. So our body mobilizes for action. Parasympathetic, right? Para, kind of like paranormal, beside normal, paramedic, beside a medic. Para means beside. And so the parasympathetic nervous system works alongside the sympathetic nervous system. But interestingly, it's the one I think is more sympathetic because it's the rest and digest portion of the nervous system. It conserves energy and rest. It slows heart rates after escaping, right, or when we're relaxing. It conserves energy, and it regulates our functions, right? It doesn't let our body kind of get too out there. So the sympathetic nervous system is our fight and flight. Maybe you can think about it that if somebody's using their sympathetic nervous system because something bad is happening to them, you're gonna feel bad, right? But parasympathetic, you're kind of just around them. You're not dealing with them, and maybe you don't have to go through that same kind of increase of mobilizing in action. However you figure it out, it's kind of a pain, so do make sure you do feel comfortable understanding the difference between the sympathetic and the parasympathetic nervous system. But to help you explain that further, right, the sympathetic nervous system operates on the 4 F's. Fighting, fleeing or fight, feeding, right, so eating, and mating. So those are the 4 things that increase bodily arousal. And I know I keep using this word arousal, and I want to be very clear what I mean by that. Arousal has a definition in the common nomenclature that we would say today that is different than what we are talking about. Arousal is this idea of kind of general body energy. So is this, right, when you're excited or right, if you just got off a roller coaster, your body is, right, your sympathetic nervous system is, right, going. You maybe are sweating, your heart's beating faster, you're a little bit jittery, you're jumping up and down. That is increasing your bodily arousal, kind of the feelings going on inside of you. That does not mean sexual arousal, right? Although sexual arousal is a type of arousal, it is not the only type of arousal we're talking about. So, right, when we say bodily arousal, it does not mean, right, what most people might think it is. On the other hand, we have the parasympathetic nervous system, which reduces bodily arousal because it allows us to rest and just digest. So giving us a little bit more time to relax. So that's the peripheral nervous system. So the peripheral nervous system, to be clear, is going to be the somatic nervous system. So the sensory and the motor neurons that, right, make us move and feel, as well as the things that make us excited and getting ready to run if we need to, versus recognizing the times in which we need to relax. So we've got movement, we've got sensing, we've got fight or flight, and we've got rest and digest in the peripheral nervous system. But in the central nervous system, we have two things: the brain and the spinal cord. Let's start off by talking about the spinal cord. So the spinal cord is the pathway connecting the brain and the periphery nervous system. So what this is almost like another information highway. It's kind of like its own axon for the body, if the brain is the soma of this, right? The kind of head of the show. So sensory neurons, as we know, they receive signals from the senses, and those get sent all the way up and into the spinal cord. And the spinal cord has these very, very long neurons, as we can see down here, neurons with very long axons. Because they don't have to switch over and do the neurotransmitters to the next neuron, the neurotransmitters to the next neuron, because it's just a single electrical signal zooming by, it's going to be the faster way to travel. So we can send these things very fast, right, to our brain and send them very fast down. Now, one thing I've been a little unclear on here is reflexes. So reflexes do exist. The reason that you don't have to put your hand on a hot stove and then think about it too long is that we have simple reflexes, or what we call the reflex arc. And so this is actually done in the spinal cord. It is the idea that there are reflexes we have that are so basic that we don't even need information to be sent to the brain to know what to do, right? If we feel our hand get if our hand gets too hot on a hot stove, which it does pretty quickly, we don't even got to think about it. Our brain does what we call a reflex arc. So instead of sending it all the way up, right, to this neuron or to the brain to make a decision, it might only end up in the right neck. I'm not really sure where this is, to be quite honest, but it's lower, right? And the idea is that instead of going up to the brain, it kind of goes, yeah, I actually know what to do. And it gets sent down. It's kind of a first line of defense. In the same way the spam filter keeps things out that don't need to be there, this is kind of like when you call a hospital or call your doctor's office and they say, just call 911, if this is an emergency. This is an emergency. My hand's on a hot stove. So my brain says, actually, we're just going to reroute you and tell you what to do instead of going through everything. Hopefully that makes sense. If not, just ignore me. Okay, so let's move on to the most important thing that we can talk about regarding, or what most people think about, and that's the brain. And actually, something really cool is I have a 3D printed version of my own brain. This is my brain based on a CAT scan I got to take about a year ago. So this that you see right here, minus the crap I can't get off it, obviously, is my brain. Now, I don't know about you, but I think that's the coolest thing ever. Right? This is what makes me, right here. Cool. Looks like this when it's not printed in gray because it was the cheapest color, and it's not covered in cerebrospinal fluid, etc. But I'll be using this for a couple examples here later on. But the brain, some fun facts about it. It weighs 3 pounds when it's covered in cerebrospinal fluid. When it's dry, it's only 3 ounces. It's like a sponge, right? It has so much going on. All those, all those ions, all the neurotransmitters, everything that needs liquid to function, right? It's like a sponge. 80% of the brain is what we call cortex. And cortex is this kind of outside aspect that we see with all of the little folds and such. This is going to be the cortex. What's in kind of the center of the brain up in here, which again, this is not what it's supposed to look like. That is going to be the central portion of the brain. But the 80% of it is the cortex, which is absent in things like fish, reptiles, and birds. And only about 1/3 of it is visible. It goes pretty deep in there. If we were to spread it out flat, the cortex is about the size of 1 sheet of newspaper or 4 pages of your textbook. So here's a textbook, right? This times 2 is how big this is. I don't know about you, that's crazy to me. That's really incredible, if you ask me. Or maybe you don't care. But either way, recognizing the fact that this thing is huge and it plays such a huge role. It's less than 2% of our body weight. But get this, it takes up over 20% of the calorie intake we take a day. It's hugely detrimental in terms of how much effort we have to put into keeping this thing. So why do we use it? Why is it so unique to humans, right? Why do we have a cortex? Why do we spend all this extra energy on it? Well, because our brain interprets and stores information, right? It sends this to muscles, glands, and organs, just like any other creature. But it has some cool things that only we are able to do. We know animals can communicate. They can, right? They can chirp, they can make noises, they can bark, what have you. But they can't do the things we do. No animal can create a computer system that can, that we know of, that can create a computer system that can let me transmit my thoughts through a microphone that somebody made into a software program that I can then upload online for you guys to learn about what it is that made this, right? That's pretty crazy if you ask me. It's the most complex part of the nervous system and allows for the richest information processing. Where our reflexes allow us to go, that hurts, kick your leg out because you have a reflex, or hey, take your hand off the stove. This allows us to take that information and relay it with other things we know and tie it into other things. You take all of this information you know, you hear what I say, you interpret what that means, you think of examples, right? I can draw you a picture in my mind. It's like, imagine this, and you're able to picture that in your mind. This allows us to do so much, but we're really bad at understanding how. So one of the first ways that we actually looked at this was this pseudoscience called phrenology, and this is just a little aside that I think is rather cool. Many of you maybe have heard of phrenology or the idea. The idea of phrenology is the idea that there are certain parts of the brain that are relatively, that are important for certain functions. So here, right, that for my brain, this way. That right here is my individuality, is what this guy says. Right up here is my firmness and my veneration, my benevolence. We can talk about how back here, this is my philoprosensitiveness. I'm not sure what that is. This is my inhibitiveness, and we can see how that's one way to imagine the brain, right? Is that each of these parts has a specific function. But is that the truth? Is that the way this works? No. This is a pseudoscience which talked about the bumps in the skull to predict mental traits. Its main heyday was from the 1810s to the 1840s. It is a joke. It is not real. There's not specific pieces of your brain that deal with, right, what are some of these, right? Memory. Well, excuse me, memory is not just some little piece of the cortex up here. Spirituality is not just some piece up here that if we touched it, we can make it disappear. Well, we know a lot more about the brain than what we knew in the 1810s. And we can break the brain into 3 different parts. And they can be the forebrain, the midbrain, and the hindbrain. So we have our forebrain, which is pretty much what you see here. Our midbrain would be right in the center down here, kind of like almost as if this was a helmet, right? It's what is in the side. And then we have the hindbrain. And the hindbrain is going to be what you can't see here, which is going to be the brain stem that comes down this way. And so let's start with the hindbrain. So the hindbrain is the most ancient and basic structure of the brain. Think about how we would have evolved. We would have evolved right from the neck kind of upward, and so we would have started with the basic things. And this is what we find in the back of the brain, the hindbrain, is we find major structures that are focused on the kind of oldest and most crucial functions of the brain. So the 3 major structures we'll talk about are the medulla, which includes the medulla oblongata, the cerebellum, and the pons. So the vital functions of this, right, with the medulla being right here, actually. So I hate this. I apologize. You can't see this. Let me see if I can. Sorry about that. I apparently had the medulla hidden. So the medulla is right here, which we can talk about right over here. So the medulla, right, and here's your oblongata because everybody seems to care about it, is going to be focused on some of the most basic functions. This is going to talk about our blood pressure, our heart rate, our breathing. And by talk about, I mean, this is going to control these things, not just talk about it. So this is where our blood pressure is kind of kept in check. So this is where you might feel lightheaded if you stand up here. It's responsible for the most basic and just involuntary functions. Beyond this, though, we can talk about some other pieces of the brains, right? The pons, which many people have not heard of. The pons is going to be the bridge between the upper and lower parts of our brain. This is where we have crossover for motor nerves. So these go from kind of our mental, maybe more like neural nerves that we might call them, to down here in the pons. This is where we're going to talk about saying, okay, so now switch that to a motor signal so that way we can run it through faster, right? And have those myelinated nerves. What's also here, they believe, is things like sleep and dreaming and arousal, which makes sense that we would need them to be in this older part of the brain. However, what's really funny about this is we don't know much about this other than people who have damaged pons. Right? We're able to study those people in kind of a case study way to understand what it is we're seeing here. So is it the case that, right, if the pons is damaged in this way, there are issues with sleep, there are issues with dreaming, things of that nature. So some of, right, issues with dreaming and sleep may come from a physical deformity or, right, some kind of either an illness or an accident that causes an issue here. But again, this might also be due to neurotransmitters and things like that. Anyways, too much. So lastly, with the hindbrain, it's important to recognize the cerebellum. The cerebellum, interestingly enough, is not on my brain in this model. But the reason it is because it's not, is because it's so dense, it's so difficult to understand. Scientists are still trying to figure out what is in the cerebellum. What we know it is this is where our fine coordinated movement comes in. And look at how kind of spongy it is here. You can tell. This means it's used a lot more and it needs to be developed very well. And so our fine coordinated motor movement, for some people, myself included, I don't have fine coordinated motor movement. It's not as fine as other people's when it comes to my writing. My writing is chicken scratch, right? Well-rehearsed movements come into play. I know that I like to throw my hands around. These are well-rehearsed movements. We do see that when you drink and have alcohol, this is affecting your brain. Here, often it makes you kind of less likely to be able to stand, be less coordinated, have slurred speech, balance issues. And that's not just if you drink, right? If you have a stroke, this is going to cause issues as well. But not only that, it's going to possibly, there are some things that we'll talk about it in class, some issues that we can see in the cerebellum as well. That the fine motor movement can be decreased, and we can't maybe do certain things without all of our cerebellum. But let's talk about the midbrain as well. So the midbrain is much, much smaller comparatively. The midbrain integrates movement with sensory input and includes what we call the reticular formation, which is involved in arousal and, wouldn't it be nice if I finished that, arousal and sleep. I don't like to spend too much time on the midbrain because it's a bit confusing and it's not really my area of expertise. And of the three areas of the brain, I think it's still the most not well understood. So this integrates movement with sensory input. So the fact that, you're able to move, and we know that, but the fact that you're able to, catch something is taking your sensory inputs, your eyes. I'm going to drop this if I keep doing that. It takes your sensory input, your eyes, where your hand is, all of this. This is stuff we can't teach robots to do yet. We're still working on having robots catch things very well. But we know a computer or a robot can calculate things very fast, much faster than we can. But stuff in the midbrain is maybe stuff that computers and robots have not yet caught up with, that we're still working on it. Within the midbrain, we have what we call substantia nigra, or in the striatum. And the striatum is involved in this smooth movement, so being able to do things kind of not choppy, right? So if we see somebody who, for instance, is a long-term alcoholic and they aren't drinking, their striatum, right? We see their midbrain, they might have shakes and they have trouble with smooth movement, so they might not be able to hold these things. And one thing that might be causing this is what we call substantia nigra, or black substance, right? Nigra meaning black, substantia meaning substance. And what that does here is it seems to be something that really focuses on movement and smooth movement. And what happens, we see, here's the striatum up here, here is our substantia nigra down here, is that people with Parkinson's, these individuals actually find lower rates of dopamine in their brain, and that they don't have substantia nigra, or the cells in the substantia nigra are dying. They're not able to be kept. And it seems to be the case where this substantia nigra is not working right and causing issues with fine motor movement. So the midbrain, what I'd like you guys to know is the striatum, and how it's involved in smooth motor movement, kind of integrating, our general base movements that we know down in the ponds with our environment, our ability to do things like catch and juggle and things like that. And that substantia nigra in the brain, which helps do this, can be causing problems for some people. And that substantia nigra is a substance that is definitely worth studying if we're interested in things like learning how Parkinson's works. Okay. So let's move on to the forebrain. So the forebrain is our largest brain region. So as you see here, this is kind of a better breakdown. We have our forebrain, which includes our cerebral cortex, our outside of the brain that we see here, and then our limbic system. Now, limbic system, don't get it confused. It does not mean limbs. We've already talked enough about movement. But we have our midbrain down here. We have our hindbrain here. We have our wow, I can't think of what this is, the cerebellum over here. And this is going to be the vast majority of our brain. And this is what has been most recently developed in terms of evolution. This is the most complex aspects of behavior and mental processes. Everything up to the brainstem, kind of up through the midbrain at the kind of more or less right here, stopping right about here. This is what's going to keep us alive and keep us moving. But what keeps us thinking is the rest of it. So let's first start out by talking about the inside here, kind of this helmeted aspect that's put into the cerebral cortex. So this is going to be the limbic system. And the limbic system integrates emotions and memory. So the limbic system is a collective name for things that are involved in emotions, memory, but also things like motivation too. It refers to a couple different structures, of which case I'm going to spend some time talking about just a few of. The first one we'll talk about is the amygdala. Now, you've probably heard of the amygdala at some point. If not, it's a common word in psychology. In the amygdala, this is a word meaning almond in Latin. An amygdala is focused on a couple different things. Mostly, we think of the amygdala as involved in emotion. And what we mean by that is how we express emotions and how we recognize emotions. So the amygdala is crucial for helping us learn to associate objects with emotional responses. Most of us can probably remember a time when we ate something and got very, very sick afterwards. This is a function of food poisoning, right? Or perhaps it was unrelated. Sometimes, for instance, me, right, I had a very strong aversion to Campbell's chicken noodle soup after I got sick one time from it. Well, what happened there was my amygdala helped me learn that response, right? It's helpful for learning things related to emotions. And one of the most powerful emotions that help us deal with and learn from is fear. So if I get sick from, right, Campbell's chicken noodle soup, and my amygdala is on, right, high alert about that, it's going to be like, yo, that's bad. I'm afraid of chicken noodle soup. I still won't eat this. And that was maybe when I was 10 years old, right? So the amygdala is going to be about emotion. Now, the other one we talk about is going to be the hypothalamus. And what I want you to notice here is that the amygdala is wrapped around, right, this big old thing here, and then we have the hypothalamus here is the purple. So the hippocampus is a seahorse. Oh, excuse me, I'm so silly. I did these backwards like I thought. The hypothalamus is our almond shape right here. And our hippocampus is this big circle that goes around the thalamus, right? It kind of, right, more or less, right, it wraps around. Now, it doesn't wrap around the brain, obviously. That's just kind of a way to think about the thalamus here. This is going to be related to the formations in new memories. It's shaped like a seahorse, so they say, hence where we get the name hippocampus, is the formation of new memories. One of the best ways you can think about this was if you saw a hippo on campus, you'd remember it. So if you've ever seen the movie Memento, for instance, right, you may have seen this movie in which case this person has trouble with amnesia and remembering things. And we'll talk about this more when we talk about memory, but that would be an issue with the hippocampus, that the hippocampus might have been damaged and might cause issue in the formation of new memories, as well as remembering possibly old memories. Beyond this, we can also talk about the thalamus, or the relay station. So this is going to be what the hippocampus wraps around, and it's going to be above the thalamus, or the hypothalamus. Hypo means below, right? Below the thalamus. The thalamus is above the hypothalamus. And what the thalamus does is we can consider this almost a relay station. So what it does is it takes the signals from our sensory organs, right, things like pain, and what it does is, right, all of those periphery nerves get sent up to us, they get put into the thalamus, and the thalamus says, okay, send it where it needs to go. And so the thalamus allows it to kind of choose the directions in which this goes, and helps us to make sense of our sensory outputs. The hypothalamus, on the other hand, which is below the thalamus and right near the amygdala, is going to deal with the 4 F's. It's going to deal with our drives that we decide to, right, what do we pursue, our motives. It's also going to work with the pituitary gland in terms of hormone secretion and temperature regulation as well. So a lot of this stuff is really important kind of in the center of your brain, but again, we're still not even getting to the idea of like our rational thoughts. We're still just trying to understand, right, at this point, all of what we've talked about in the brain, just trying to understand how it is we are able to remember things, get things done, and move, and we're not even thinking about really right, actions or higher order thinking. But we can move on and talk about that as well. So let's talk about the cerebral cortex for a minute. So the cerebral cortex, again, is this outer hemisphere of the brain. So this is going to be the outer surface of our cerebral hemisphere. This is going to be what we can see here, all the squiggles and lines and folds in our brain. Now, it has some different anatomical areas. So first of all, let's orient ourselves to my brain. So my brain is, right here would be my eyes, so it would look like this in my head. Perfect. So here's our front, and I think, okay, see, I have it the right way. So the frontal lobe is going to be right up front by our kind of top of our head. So our frontal lobe, we're going to have our temporal lobe, parietal lobe, and occipital lobe. So I should actually just put all these up. This makes more sense. So frontal lobe is going to be this stuff right here. And this is going to be this where we find our higher order thinking. This is going to be the last portion of the brain that's really developed. And when we talk about our occipital, or excuse me, our frontal orbital cortexes right here, right under the eyes. This is the last part of the brain, the most recent developed part of the brain right here. And this is where we have the highest types of thinking going on, because we've got all the rest of our brain that's been built up over the years to figure out everything else. Now we can start focusing on higher order thinking, decision making, right? Doing complex tasks and understanding all of these things that make us human. So the frontal lobe is up here. We can also talk about the temporal lobe, down low, down here. Temporal lobe, right, meaning near the temples, so near, right, the ears, as it makes sense, right here. This is going to be where we talk about things like hearing, memory, emotion. Some of the language stuff is here as well. The parietal lobe, which is kind of this back half over here, not everything, is about processing information from the body's senses. And we'll talk about the somatosensory cortex here in a minute. The last one we'll talk about, the occipital lobe, right back here, is really where our visual cortex is. And that takes up a lot of energy in our brain. So the occipital lobe, right, occipital, ocular, eyes, things like that, that's going to be focused on visual recognition, visual understanding. So quickly moving back to the parietal lobe for a second, this is where we have our somatosensory cortex, where we have things like our sensory cortex, our motor cortex, and our association cortex. And what those look like here, right, are some different places in the brain where, right, these are going to be focused on very specific things. So the visual cortex, right, we know is in the occipital lobe. So that's a little different. But our somatosensory cortex in the parietal lobe is going to be focused on things like touch and pain and temperature and dealing with that. Lastly, we can talk about the audio cortex, which unsurprisingly is near the temporal lobe, right near the ears. Makes sense. We don't want to travel too far with that, and it deals with sound. So with the association cortex and the somatosensory cortex, parts of the cortex that aren't involved in these things are dealing with complex cognitive functions. They often combine information from more than one area. But what happens when we can't or we have issues? Well, we have things what we call aphasia. So aphasia might be difficulty in understanding or things, for instance, in producing speech. So two types of aphasia that your book talks about, and I want to talk about, are Broca's aphasia, which is important for speech production, and Wernicke's area, which is important for the comprehension of speech. So Broca's area is actually in the forebrain up here, in the frontal lobe, and the Wernicke's area is down in the temporal lobe, more focused on right near the hearing and understanding of this. One way to remember these is that Broca's area, it's dumb, but a friend told it to me once and it's never left my brain, is that if you can't speak, part of your brain is Broca. Hence, somebody with a Broca's aphasia is not going to be able to speak very well at all. Whereas somebody who has Wernicke's aphasia is having trouble comprehending speech. They're able to speak, but it's without meaning. So they might say like, store chair went today. And you're like, what? Right? So they're speaking, but it doesn't make much sense. So recognizing that there are lots of parts of the brain that are dealing with other things, and sometimes if some of them get damaged, we can see these results show up. The last thing I want to talk about is the brain's hemispheres. So as you can see down my lovely brain, we have my two hemispheres, which are very clearly noted based on this kind of trench, you might call it. And I don't know how you can actually see it, but it's rather deep in there. It goes for about half an inch or so, where there is a space between the two halves of the brain that they don't touch, right? And you may have heard of things like the left versus the right hemisphere. I am the left brain. I am a right brain person. Well, not quite. So with the left and the right hemisphere, they each act on different things. The left hemisphere deals with language. It also deals with the right side of the body. The left half of the brain deals with the right side of the body. The right half of the brain deals with the left half side of the body. Very confusing. Not sure why they did that. Doesn't make a lot of sense to me, but it's how it works. The right hemisphere, not only does it deal with the left half of the body, but it also controls some nonverbal information. Things like visual recognition, emotion, perception, things of this kind of detail are more done on the right side of the brain. That does not mean they're only done on the right side of the brain, because some functions are what we call lateralized, go between them. One thing I didn't mention is the corpus callosum, which you can see technically split here, right? It's been severed in this example, but the corpus callosum would be down in here, and it would be a bridge between the two sides of the brain. Corpus means body or kind of structure here, and callosum means colossal, big. So really what this is this is a big piece of brain that cuts through that hemisphere and goes between the two. And so the corpus callosum lets our sensory and motor processes be available in both halves of the brain. Our language, which is typically in the left side of the brain, is typically there, but there are some things that deal with the right side, and nonverbal language, which is typically on the right side, does have some stuff on the left side, but for the most part, those are kind of separated. When we think of our right and left hemispheres being the same, the left does have slightly better logical and language skills. The right has slightly better spatial and artistic and musical abilities. And PET scans do show these differences. However, the individual variability and the precise nature of the degree of lateralization is completely not understood, and these hemispheric differences should not be exaggerated. So, recognizing that our brains do have specific things that go on in one half versus the other. But if you're a young child and you're missing parts of your brain, it's actually the matter where you can still get through life where your brain, right, if you lose half your brain will reconfigure itself to be done with everything on one side of the brain, which is very, very cool. But I have spent way too much time talking, so a couple quick announcements. Keep up with the materials. If you have questions, stop by office hours. But other than that, I hope you all have a great day. And if you need anything, let me know. Otherwise, enjoy.